Let's notice all the important tests to confirm the presence of amino acids and proteins. See, there are mainly 5 different tests that we will perform. First one is called burette test. Burette test is the one where we use copper sulfate in an alkaline solution, correct? And by chance, if there is a positive test happening, that means that solution will convert into pink violet to purple violet color. Pink violet to purple violet color. Basically, what is it exactly testing and why is it called burette test? Now, this compound, although it is not an amino acid, it looks like amino acid. Why? It has an amide chains. It is a peptide bonds, correct? It has peptide bonds. It is not amino acid, but it is having a peptide bonds. So this is taken as a basis. If this compound, when you combine with copper sulfate, is giving a positive test. If it is giving a positive test, what does that mean? It means it is connecting with copper, becoming a complex, and that complex is giving that color. Okay, which means what is essentially observed here? See, here what is observed, the and how the violet color is been observed here. Okay, see, when it comes to burette here, if you notice there is a lone pair present here, lone pair present here. Of course, there is a lone pair present here also, but it is mostly delocalized all the time. So you can see I should have NH2 and NH2, at least two of them, correct? Suppose here it is interconnected, let's say. So one from here, one from here. If it connects this way, and here, one from here and one from here, it connects this way, then it becomes violet color more precisely. Got it? So which means what are we expecting? That we should have NH2 type of groups, but at least two of them should be present. Okay. So which means the burette test works the best if you have combination of any two of the following. That means these two, these two, or these two, these two, these two, or these two, like this. Any two being combined. Minimum two. More than two is perfectly fine. Correct? Which means this test is not positive if you have only amino acid, single amino acid. No, you should have peptide linkages. That means you should have at least two amino acids, three amino acids or so, correct? So for all the uh, proteins, this will give you positive test because proteins are polypeptides only. So minimum is dipeptide. So if you have a dipeptide, tripeptide, tetrapeptide, for all of them, you will get positive. If you have only amino acid, it is like a monopeptide. Okay, then there is no complex formation here. Okay, see next one. Next one is ninhydrin test. The ninhydrin test is a test for amino acids and proteins with free NH2 group. This is with a free NH2 group. Okay, if amino acids are detected by ninhydrin, all amino acids will give violet color product with ninhydrin except proline. Okay, what does that mean? See, if this is an amino acid, I take 2 moles of ninhydrin. This is ninhydrin. So basically ninhydrin will take hydrogen and OH from here, convert into amine, and it will perform 2 times. So 2 times OH will go away and gets an N connection here between these two. Okay. So this nitrogen completely comes in between the two ninhydrins, and this part will be lost as an aldehyde group. Clear? Aldehyde, and of course whatever the carbon which is present here, that will come out as carbon dioxide. It will get oxidized. The point here is to understand that the ninhydrins will start getting colored formation. So it will give purple blue color. Okay, it will start forming purple blue color. Understood? Instead of primary amino acids, I mean primary amino acids, if you have a secondary amino acid like proline, it will give yellow color instead. Instead of giving purple color, you will get yellow color. Okay, that's the difference. So ninhydrin is used for identifying any amino acid. But what is the condition? That it should have a free NH2 group. Correct? Xanthoprotic test. This test specifically is given positive for tyrosine, tryptophan. Tyrosine, then tryptophan, and in fact, phenylalanine will also give the test, but it responds very weakly. Okay, what does that mean? This is tyrosine. Okay, this is tryptophan. Have you noticed in both of them, the ring is activated. The benzene ring is activated. Okay, and on an activated benzene ring, when you add concentrated HNO3, nitration will take place. You can see here the para is already occupied. So at ortho position, nitration will take place. Similarly, in tryptophan also here with respect to this, para is open. So at para position, you'll get nitro. So both tyrosine and tryptophan will give you positive test. Okay, phenylalanine will be very, very weak. So that's why we do not consider much about it. Okay. So xanthoprotic test is specifically designed for tyrosine and tryptophan. So how do we know that it is making a reaction? Once it forms this nitro compounds, they appear to be yellow or orangish color. Agreed? So if I see yellowish to orange color, that means it is confirmed that there is a presence of tyrosine or tryptophan in the chains. Clear, beta? Right. This is remote. Sakaguchi test is not there for you. Next, Millen's test. Millen's test is when you react with mercury, Hg2 plus. Okay. So, but to do that, first we need to make a uh, oxime. How do you make an oxime here? You take, any NH2 group, open NH2 group, that when you react with HNO2 will convert into nitroso group here. Okay, it will convert into this nitroso group, and that will appear like an oxime, and that oxime gets connected with mercury 2 plus, and which gives reddish brown color. So you might ask, sir, ये सब learn करना है क्या? नहीं, ये सिर्फ टेस्ट पता करना है आपको. What is the test? What is the color? And what is it valid for? That's it. Okay. It's only for your reference. Clear? Hmm. So you can see a reddish brown coloration of precipitate indicates the presence of tyrosine. Again, this is specifically designed for tyrosine because only when you have such active ring, you will have NO getting substituted. Sanger's reagent. Sanger's reagent basically is 1-fluoro 2,4-dinitrobenzene. Okay. What would it do? See, it needs to have an N-terminal amino acid, and the N-terminal has to be NH2. It cannot be 2 degree or so. And from here we can have double bond NH, and this can keep on going, some kind of a amino acid sequence here. But on the N-terminal, there has to have NH2 group. And then you take that NH2 group, react with DNFB. DNFB is dinitro fluorobenzene. Okay. When you do that, fluorine can be removed out and nitrogen can be attacked here. Okay. And once it comes out here, then hydrolysis. On hydrolysis, this will break. Okay. All the amino acids will break. And you will get this part, and this part. Clear? So from here, you will know, because these two got separated here, based on the molar mass, now you know which was present on the N terminal, which amino acid is actually present on the N terminal. So Sanger's reagent is mainly used to understand the sequence. Okay, so when you react with mercury, HG2 plus, you will get this part, this part, and this part. Clear? So from here, you will know, because these two got separated here, based on the molar mass, now you know which was present on the N terminal, which amino acid is actually present on the N terminal. So कि ये सीक्वेंस कहाँ से स्टार्ट हो रहा है, which is that amino acid from where it is starting from. Okay, clear. So these are all, studies that are being done in, forensic departments. Got it? Clear, especially for DNA identification and all of that. Okay, and protein identifications. These are all the tests that they do. Sanger's test is one very prime test to understand the protein chains starting from what sequence. Clear? I guess the tests of proteins as understood. So these are the four main tests that you need to know. Next is about test of carbohydrates. Okay. The first main test is Mollish test. Mollish test is mainly used for identification of all carbohydrates. That includes monosaccharides, disaccharides, polysaccharides, and so on. Okay. So basically, what we do, we add sulfuric acid to hydrolyze all the glycosidic linkages to yield monosaccharides first. So we want all of the oligosaccharides, okay, or polysaccharides which are present. Let them break down into, small monosaccharides first. That is the first thing we'll do. And once you get all the monosaccharides, let us assume that the monosaccharide mainly is made up of glucose, right? And if this glucose has been further kept in concentrated H2SO4 with a controlled form, then here this O will attack, this bond will break because it is an hemiacetal bond, correct? Hemiacetal will break, converts into an aldehyde, and the ring suddenly changes into a five-membered ring. So the same glucose will convert into a new aromatic ring form, which is furfural. The name is called furfural. Got it? This is basically a furan. You know that this is furan, correct? Furan with an aldehyde. This is called furfural. Clear? So 5-hydroxymethyl furfural. And now you leave it for some time till the furfural is formed. Furfural is a colorless solution. And on that colorless solution, you add beta naphthol. Okay, slowly start adding beta naphthol. Clear? And as you add beta naphthol, I mean, the acidic medium is still there. Then what happens? From this para position, it will attack here 2 times. Okay, because you know that aldehydes can undergo uh, electro electrophilic aromatic substitution. So it can happen on para position from here 2 times. So 1 mole from here, another mole from here, 2 times you can get this group. Okay, and that you can dehydrate with this OH. You can make it as double bond O and dehydrate. This H is not there here. Yeah. So this dehydrated form is what will appear as a purple or a violet color. Clear? Mollis test is important in terms of the in terms of the structure also. What exactly the reagents used. Okay, let me repeat again. You take any oligosaccharides or polysaccharides, convert them into monosaccharides. Once you get monosaccharides, okay, you will do a controlled amount of dehydration to convert into furfural. And once you get furfural, when you react with alpha naphthol, it will turn out to be a purple color complex. Done? This is called Mollish test. Next one is Barford test. Barford test is mainly used for differentiating monosaccharides from disaccharides. But to do that, we require to use a mild acidic condition. So if we use a mild acidic condition, correct? Any form of, any form of monosaccharides we have, they get easily reduced. I mean, they get easily oxidized, and copper will get easily reduced to form red precipitate. Okay, when you have mild acidic medium, you know that this is nothing but a Benedict test, correct? Failing solution or Benedict solution, but failing and Benedict are taken in basic medium. Now I'm taking in acidic medium. Got it? Right. See, How are we differentiating with disaccharides and monosaccharides is based on the speed. If it is disaccharide, it will take more than 2 minutes. Okay, in fact, more than 3 minutes, sorry. Okay, so 10-15 minutes like that. But if it is monosaccharide, within 3 minutes, we will get the precipitate. That's how you differentiate whether it is monosaccharide or disaccharide. Okay, clear, beta? Next one is Shelvinov test. Shelvinov test is mainly used to differentiate fructose from glucose, or in a way, ketose from an aldose. How to differentiate ketose and aldose? See, this test mainly relies on the principle that ketohexose are more rapidly dehydrated than aldohexose. So, in fact, ketones will have more easy formation of enol and they can get dehydrated much faster to give furfural compared to aldehydes. Okay, so this is what happens. First, you can have fructose. Fructose on dehydration will convert into this very easily, and then sorry, this is the furfural. You react with 2 moles of resorcinol. Okay, so resorcinol again reacts with the aldehyde form of it. Here, this is the aldehyde form 2 times, 2 moles, and 2 moles of this resorcinol. From meta position, again it gets dehydrated and finally converts into cherry red precipitate. This is called Shelvinov test. Clear with Shelvinov test? Right. Bell's test is not there for you. Next, osozone test. Osozone is something which we learned already in carbohydrates. Let me just repeat it. Basically, glucose, mannose, fructose, these three will have same structure from third carbon onwards. The differential part is only about first and second carbon, correct? And osozone mainly forms and first and second carbon. And glucose, mannose, fructose will form the same osozone. It will be needle shaped, correct? So you cannot differentiate glucose, mannose, and fructose. While maltose will give you a sunflower shaped osozone, lactose will give you, a powdered puff shape osozone. Got it? So osozone test is mainly to observe whether it is maltose, lactose, or glucose. That's it. Okay, based on the shape. Clear? Next is Benedict test. Benedict test is something which has been given by all the aldehydes, not by keto groups. Okay. But yeah, if you have fructose is a hydroxy keto group. So if you have alpha hydroxy keto group, that will still come out to be positive value. So you can see glucose, galactose, maltose, fructose, they all will give you positive test. Which will not give positive test? Sucrose and starch and cellulose. These will not give positive test because they are non-reducing sugar. Okay. Benedict and failings are exactly same. Next, Tollens. Tollens is something which you have learned it already. All the forms of aldehydes can give. Okay. Only difference between Benedict failings and Tollens is Tollens can give positive test with benzaldehyde and glyazole, but these two will not give positive test with benzaldehyde and glyazole. Okay. Done, beta. Right. So iodine test is mainly used for starch, only for starch. So in the starch, if you add iodine, it will turn into deep blue coloration. Okay. So that deep blue coloration is observed when you add iodine. So that refers to the presence of starch. That's it. Okay. So these are all the tests that you need to know. Clear? Okay. From the test point of view, what you need to learn from biomolecules test is carbohydrate test and amino acid test.